Hello everyone, welcome back to the session on rodent behavioral models and setups. So let's look at a little bit in detail how exactly these kind of markerless techniques are going to help us. So first and foremost is that you need to sort of look at the anatomy of the rodent's forepaw, and you can see the digits here. All right, there's a little finger, ring finger. So uh, like just like in humans, there are joints. This is distal interphalangeal joint, proximal interphalangeal joint, and the metacarpophalangeal joints. So if your camera is good, it will capture the limb anatomy very well, and very crisp image can be obtained where you can put the markers and label it in your algorithm. All right. So you need to tell the software, okay, this is the tip of the digit, and that is the proximal interphalangeal joint, that is distal interphalangeal joint and the proximal interphalangeal joint, or any other common terms you want to name it. So each and every digit are marked, and then the data is trained. So this is very important, because once you train, okay, this is what the digit and the joints are, then when this moves, automatically the software say where is the position of this particular tip of the digit. So let's take an example of this particular point in space. If this moves, the software detects the trajectory that it has moved there, and then it has grabbed the pellet, and then it comes back. So that entire movement is fed into your training algorithm, and then it is really easy to capture the path that is taken by the digits which you have just marked. All right. So that this is another example where you can train the algorithm and name it as the reach, the grasp, and the retract. All right. So these steps can be trained based on the movements of the digit that those are happening. So you can see how the reach is going to happen and the grasp is going to happen before it is going to retract outside the retract the limb into the setup box. So in each step you can say whether it is missed or slipped or completely dropped. So if the reach has happened, then the next step is obviously grasp. If the grasp has happened, the next step is obviously retracted. If any of these steps are missed, then you can name as miss, slip, and drop, and finally into the success if it retracts completely and then feeds itself. So before that, if the foot pellet drops, then goes into the drop. If it fails to retract, then it is, you can call it as slip. If it fails to grasp, then you say it is missed the pellet completely. All right. So based on the markers that you have chosen on the individual digits, these movements will be captured, and then you can train the algorithm as following. All right. So that's one way of looking at it. So and this is the extracted digit and hand trajectories. So as I said, how those digits, for example, that is the point that you had chosen on distal interphalangeal joint. So where is that point moved? It has taken that path and then taken the foot pallet there, and then it has come back here. All right. So that is the trajectory that is taken by the extended digit. Similarly, if there is a flexed digit, then it can study the movement of the flexed digit, for example pronation and supination. So how is that happening? You can look at it individually based on the marker that you have placed virtually in the software. All right. So then you really don't have to have a physical marker on the rodent body, and that's the whole point. And then you can look at it as the end point of the trajectories. So it has started from there and then it has reached there to grasp it. So you can define your end point as well. If the hand goes out somewhere, maybe that can be ignored, or that you can train it as miss, where the rat misses the target completely and then goes out. When the trajectory goes out, you can say it has missed. Alright, so based on the retraction, that is the trajectory backward, if that is incomplete due to stroke, then you can easily make out that the path has changed as opposed to the trained data before your stroke surgery. So that's where things gets bit easier to analyze, and the results are automated, and the output is also automated, where you can easily look at the trajectories itself and make out the percentage difference in the performance of this particular behavior. So you are going to get much more objective answers to the changes in the posture, changes in the retraction trajectories. So all that are going to be a bit more objective, and it's easier to analyze, and these are more valuable or then authenticated than the visual inspection, which can go wrong or which can be really subjective. Alright, so it might dilute your own data if it is not really objective. If not to this extent, at least using the markers we need to analyze it. So that's a video which explains the entire task of the head fix cue multi-step prehension. You can see the disc has moved after the cue, and then that's the grasp that is happening, and the sup, and then it's into the mouth, and then the forelimb comes back into its position. So that's the beauty of this particular, and then you have there the time scale of 62 milliseconds for that entire task. I will play it again. You have marked the point on the forelimb, so and you call it as a DIP or the the distal interphalangeal joint, and as the trajectory progresses, then you can break all that into different aspects of the movement. So that's the importance of analysis. So there's another so that is one trial. Similarly, if you conduct multiple trials of those, and then this particular software, like this is the example of deep club cut, there are many other software which does the similar kinematic analysis of the path taken by the movement, that is by the this particular task, where you can see in this video, I will play it. You can observe that the entire trials has been averaged to create this particular trajectory. Alright. So that the darkest one, the dark red, is the most validated trajectory, which is the average of the entire number of trials that was done by this rat. I will play it again. You watch it. Blue is the ongoing trajectory for the different trials, but the commonest path that was taken is shown in the red. So not only that you are able to observe individual steps in different trials, it also gives the most common path that was taken by the rodent. So if at all you are going to compare it with the post stroke analysis, it is very easy to have a normal distribution and then capture all the trials that comes under the curve, and it is very easy to have a standard deviation for any of the behavioral analysis that you are doing. So, it is very easy to even get in the statistics into the behavioral task and make sure that the entire behavioral analysis is validated. Alright. So, and then from here onwards you will be able to make out whether this particular behavioral test has given any answers that you are trying to achieve through that particular experiment. So, another task that we can actually use it to compare is this lever press task, where you can see in this video where there is a automated paw tracking. This is what I was saying. Instead of putting this is mostly like a marker where the right paw is tattooed with the red ink, and the red channel was then thresholded through the centroid. Then you can see as soon as it presses the lever, there is this analysis of the amplitude of movement that is happening to the marker that you have placed. Alright. The marker is something we have placed, and then you capture the darkest or the brightest point in that marker, and then you capture the video and then try to analyze it and see what sort of trajectory the rodent's forelimb has taken. And if there is any deviation that happens, it is very easy to pick it up. So obviously with the number of trials you will see number of waves that are taken. So this is another different way of looking at the kinematics of the trajectory that the rodent's forelimb has taken. Alright, so it is pretty important that different tasks can be used, whereas if I can play the video again, you can see that mostly the shoulder and elbow movements are really dominant as compared to the distal forelimb. Okay. So the movement that is occurring is pretty gross in the shoulder and elbow, whereas the finer movements are not that much. So it is sort of pressing, and then you can even see that the other forelimb also is coming into the role. So that is where it becomes little gross as compared to the V-shot test. But the advantage of this particular lever press task is that there is always a noise which is introduced by the chewing movement in the V-shot test. So as I said earlier, if your experiment involves electrode implantation and you are trying to do electrophysiological analysis where you are recording the neural signals, you want the noise to be minimum. Alright. So if there is a chance where noise is going to sort of alter your signals, then it is always better to use a task where such chewing noise can be avoided. So there are going to be some trade off either in terms of the movement that you are going to observe or some signals that you are going to acquire. So those trade offs are something you will have to discuss and analyze it, and then finalize which is the task which best suits your question, the research question that you have taken up, and then which is the task which is closer to proving any of your hypothesis. So ultimately once just before we are going to do the stroke surgery, you are going to train the rat, you induce the stroke, and when would you really know whether the stroke has really manifested in the subject that you are going to observe with various behavioral analysis. So this is one video which is very clearly see that the limb here is pretty much rigid and is not even really moving, and most you see that there is a circumduction movement that is happening because of the paresis on this particular forelimb and hind limb. So when it tries to move, it end up in moving in circumduction movement only because there is hemiparesis in the left half of the body. It is only the right forelimb and the right hind limb is moving, so it end up in moving in a circular motion. So if the forelimb is really paretic or paralyzed, then none of these tasks are going to happen. So that's a prerequisite. If you are really going to have if the rat is going to have a full blown stroke, then it may not be even be fit to subject to this particular task. But you need to remember, in stroke there is always some recovery that is going to happen. Alright. So whenever you are going to choose the rat after the stroke surgery, If it is for a translational neural engineering, or if it's a sort of an interventional setup that you are trying to look at, or interventional experiment wherein you are going to introduce some neural stimulation or physiotherapy or the combination of the two, and you want to see the improvement, then you need to choose a task which has less than 60% of success rate. That is very, very important. There are many rats which will recover and then go to have 80% of the success rate as compared to its pre-stroke analysis. Then this is going to give you a falsely positive result that your intervention has helped, whereas in reality that is the natural recovery that is going to happen in any stroke model that you consider. So that is the whole point here. One need to remember that if you are trying to look at the recovery of the rat due to some intervention that you are doing as part of your research, you need to make sure that the recovery is in fact due to your intervention and not because of the natural recovery that is going to happen. So how does this natural recovery of stroke happens? Maybe by the plasticity in its brain itself, where there are new neural connections that can happen over a period of few months, or maybe new vascularity that are recruited. For example, if that is the stroke area where the vessel is blocked, it might recruit new vessel, a new blood vessel supplying the same area, and those neurons might recover on its own. And that is the natural recovery, either by the neuronal rewiring or by the revascularization. Natural recovery can happen. So you need to keep this always in mind, because this is going to be a major confounding factor in your research if you are trying to look at some intervention which is going to benefit the paralyzed limbs of the rat. Alright, so that is one point I wanted to really highlight. So that comes to the end of the stroke model and the behavioral analysis of that particular model. So all I am trying to emphasize is that try and use this as a basic model wherein you can sort of create many other models. For example, be it epilepsy model, be it a spinal cord injury models. In any model that you are going to create using the rodent experiment, these are the ways that you can really analyze, and these are the various setups that are available. So unless you know that these things are at your disposal, you may really miss out on many such advanced technologies that are available at your disposal. So that is pretty important to really understand and remember. So in next session we will deal with the Parkinsonian model, which is very commonly used model, not just for therapeutics, nor it's also really important to understand the deep circuitry of the brain. So Parkinsonian model is another model which has been studied extensively for deep brain stimulation strategies and many such therapeutic interventions. So I guess we have selected that model as well in order to give you a good comparison with the stroke model as to how complicated that model is and what are the things that we can learn from the behavioral setup that is used for Parkinsonian experiments. So thank you all for your kind attention.